So we're going to start with chapter 1. And biology. Biology is the study of living things, which is a huge subject. So they talk a little bit about that and how everything living is very diverse, of course. So in order to try and organize all living things, we can split them into 3 categories. We call them domains. Make sure you know these. This is a short answer question on the test. Okay, so they are bacteria, archaea, eukarya. They're formal names, that's why they have them capitalized. I'm not going to take off for slight spelling errors. Has to be close enough I know that you know what you're talking about. And so bacteria, what are bacteria? Germs. Okay, so that's typically what you guys think of as germs, bacterial infections, etc. Streptococcus, staphylococcus, bacilli, that type of thing. So that's what we typically know. Archaea, that one might be more difficult. Does anybody know what that is? Okay, so these look like bacteria. And for a long time, up until at least the 90s, because this is new since I graduated from undergrad, but at least till then, they were still classified together in one group because they look so much alike. By accident, they found that the DNA was very different. Archaea are usually classified as extremophiles. These are single-celled creatures that many times live in very extreme environments, like something that's really salty, something that's very high or low pH, high or low pressure, high or low temperature, that type of thing. Places where we wouldn't expect things to be able to live. So by accident, for example, they were drilling out some rock cores somewhere, wanting to look at the microscopic structure of the rock crystals at high pressures to see what was happening to them. They stumbled on archaea type organisms. Would have never thought that anything could be living down there, but there is. So in terms of the difference physically, you won't see a big physical difference because they're single-celled. So they, on the outside, they look a lot like bacteria. But on the inside, their DNA is much different. Bacterial DNA is in a circle. They have just one chromosome. Archaea DNA is more like ours, where it's linear and they have separate chromosomes. So evolutionarily speaking, archaea are closer to us than they are to bacteria, even though they look much more like bacteria. Okay. And then the third one, eukarya. If I asked you to name living things, probably almost all would come from eukarya. So eukarya are what we call eukaryotes. I'll get to that in a second. That's where we are. And we belong to the domain eukarya. All plants, animals, fungi, little things called protists, they all belong to the domain eukarya. Before I get to the kingdoms then, let's talk about the difference between eukaryotes and prokaryotes. Okay, so eukaryotes are all of the organisms from the domain eukarya. They belong to eukarya. They have what we consider a true nucleus. That's like what defines them. They have a true nucleus. What makes it true? It has its own membrane. That's the difference. They have a nucleus that has its own membrane. There are other things I could tell you, but that's really what is the line in the sand, so to speak. That's the definition for a eukaryote. But other things, they have other internal membranes. They're usually bigger, much more complex, lots of organelles. So later when we talk about cells, we spend a lot of time mostly on eukaryotes because that is what we come in contact with visually most of the time. So us, plants, animals, etc. The other type of cell, the other type of organisms are prokaryotes. If you're not one, you're the other. So you're either a eukaryote or a prokaryote. Prokaryotes do not have a true nucleus. So no true nucleus. What that means just is that there's no membrane on the inside. Prokaryotes don't have any internal membranes. Prokaryotes don't have any internal membranes. Okay, so of the domains, eukarya is full of eukaryotes. Bacteria and archaea, those are the prokaryotes. Prokaryotes, as compared to eukaryotes, small, very simple, very few organelles, always single-celled. So that's what I mean by small. Bless you. Where eukaryotes can be single-celled or multi-celled like us, and prokaryotes are always single-celled. And then you'll notice that they have 6 kingdoms. I don't recognize 6 kingdoms. That's something that is still discussed in science. The reason why they count 6 is basically because the old system, before we had domains, was only kingdoms, and then we subdivided from there. And there were 5 kingdoms, where the kingdom Monera had both bacteria and archaea. But that's because we didn't know there was a difference. So once they found that difference, instead of, well, in one view, they decided that we should just separate that out and have now 6 kingdoms instead of 5. But then in another view, they decided, well, but bacteria and archaea could be subdivided more, where 4 out of the 5 or 6 kingdoms are really all eukaryotes. So it would be more proper 3 domains. Never used this before, so we'll see what happens. Not sure what that means. All right, so I prefer there to be 3 domains, and then the domain eukarya is split into 4 kingdoms. Those 4 kingdoms are Protista, fungi, plantae, and animalia. 3 of those, I'm sure you guys know what they are. Plantae is what? Plants. Animalia, animals, including insects. Fungi. Yeah, molds, mushrooms, funguses. Okay? And Protista, those are kind of like everything else. They are typically single-celled eukaryotes. Most eukaryotes are multi-celled, but Protista, they are single-celled. So they still have the true nucleus and the more complex cells, but they're only made of 1 cell. Questions so far? And here it is in a picture. So they're just showing you the 3 domains and the 4 kingdoms of eukarya. If you're looking at 6 different kingdoms, then here they are, where these first 2 would be, this is archaea, this is bacteria, this would be Protista, and then down here, of course, animalia, plantae, fungi. All right, more importantly, properties of life. Okay, so what makes something living? Can you name some? Somebody asked you what makes the difference between, say, your dog and the chair. Why? How do you know? Breathing, moving. What else? It can, good. Response to stimuli. What else can your dog do that a chair can't? Yes, response to stimuli. What else? Eat, breathe. That means they require nutrients. What else? They use energy. What else?
Reproduction.
Good. Reproduce. Okay, good. You've got almost all of them. Homeostasis is another one, but we're going to list them here. Okay, so they start out just by saying all living things are complex. They have movement of some form and response to stimuli, but down at the bottom they say whatever it is that you list, it has to be exclusive to only living things. You cannot say that same thing about something that's not alive. Okay, so then they number them. So this is nice. Anything that's numbered makes a really good short answer question. Make sure you know these. Okay, well, number one, anything that's living is made up of at least one cell. That's what they're saying here. It has to be made of at least one cell. So automatically, this excludes some things that kind of straddle the living and non-living. Viruses. Technically, by this definition, cellular organization, viruses are non-living because they're not made of cells. They are many times described as proteinaceous particles. They are DNA with a protein coat, something like that. The reason why they make us sick is because they can't reproduce on their own. They have to hijack our cells in order to reproduce. So that's the second thing that makes them non-living. Number two, metabolism. This is where you guys told me they breathe, they eat, they require energy. That's what metabolism is. If they need nutrients, they also produce wastes. They require energy. Your chair doesn't require energy. Number three, homeostasis. Homeo means same. Stasis means state. So this is same state. That's how it translates. What this means is living things keep things inside their cells steady. They keep things inside their cells steady. For example, it could be for things that are warm-blooded, temperature. Human temperature should be what? Close to what? 98.6 degrees Fahrenheit or 37 Celsius. Okay, it's not going to be exact for every cell all the time, but it should be close to that. pH. Our blood pH in humans should be at 7.4. And it's always going to be, unless you're really sick, it's going to be pretty close to that. For other living things, they'll have some other maybe typical pH. And certain groups of cells have their own typical pH. In your stomach, the pH there should be 2 or 3. In your small intestine, it goes above 7, so between like an 8 and a 9. And it's the homeostatic mechanism keeps it that way. So your stomach's supposed to be a 2 or 3. If you decide that you're thirsty for milk and you drink a whole bunch, milk is above 7. It's going to shoot your pH in the stomach up. Your stomach responds by the cells producing more acid to bring that pH back down. Things that are single-celled, if you think about it, they just have to maintain it inside that cell. But many times, these are in contact, say, with an environment of water. That's going to be difficult for them to do, considering how water moves in and out of a cell all the time. We'll talk more about that later, too. Growth and reproduction. Okay, so you guys told me they could reproduce, but living things also have the capacity to grow. An individual cell can grow, but also the cells can reproduce. So you get a paper cut, your body repairs itself by those cells dividing until it's all healed and repaired. A starfish can grow a new leg if one gets cut off. You can actually get 2 starfish from one if the center of the body gets split. Then each half would grow the half it's missing. That's kind of cool. There are some things that are alive. We know they're alive, but they cannot reproduce. So they may not meet this one. They can grow, but they can't reproduce. Can you name one? You'll know when I say it. Think insects. How about bees? Think about the worker bees. They don't reproduce. And sometimes they're called drones. Their job is simply to do the work. So you have one female that reproduces, the queen, right? And then there are select males that reproduce. All the rest of them are typically males that do the work. Same thing with ants. They have that same type of mechanism set up in their colonies. So there's going to be some exceptions, but overall, most living things can reproduce. In mammals, a mule is sterile. And some of you might know that, some of you might not. And so if you want to have or get another mule, you can't breed your mule. You have to breed a horse and a donkey. And it goes fine, except that mules are sterile. Number 5, heredity. When living things reproduce, they have the ability to pass on their traits. That's what this means. They can pass on their traits. They do that with genetic information. Most of the time we're talking about DNA. Probably more than 99% of the time. Occasionally RNA can be used for genetic information, but almost always we're talking about DNA. Make sure you know those 5 properties of life. Okay, the organization of life, I don't hit this too hard on the test. A lot of this first chapter is just kind of introduction. But here they're talking about how living things are definitely organized in terms of how they're put together, but also how they interact with each other. Okay, so that's what they're going to go through here. Like, you can have a group of cells that all have a similar function. We call that a tissue. And then you might have a group of tissues that have a similar function. That's an organ. And then a group of organs for an organ system. And then all of those and tissues together to make the organism. And then even as you go to separate organisms, there seems to be some organization in that they're organized into separate species. And then if we look above that into one area where there's a bunch of them living together, they might be organized where you have bee colonies, ant colonies, you have some organisms that live in the trees, and they may all have some similar features, while you'll have others that live, say, in the water. They have similar features. So living things, no matter how we look at it, tend to be very organized. Here in pictures, they're just showing you that organization, where if we start at atoms, we can put those together through bonding, you get a typical molecule. This one happens to be adenine. You'll hear that multiple times as we go through the semester. Adenine is very important for ATP. That's energy in our cells. It's very important for DNA and RNA structure. This macromolecule is DNA, of course, as it's listed here. Macro is just a big molecule. And then if you have a bunch of molecules together, they'll form an organelle. This one happens to be a nucleus in a eukaryotic cell. It's not alive yet, though. It's not until we get to this next step, a full cell, that we can say that we have something living. Because remember, a cell is the simplest living thing. It has to be at least a cell. That's that very first property of life, that all living things are made of at least one cell. This happens to be a neuron. It's a type of nerve cell, so you'll find these in your brain. When we talk about nerves, they're actually long parts of these nerve cells called neurons, called the axon. Some axons are longer, some are shorter. It depends on, what we're talking about, where at. Here's a bunch of nerve tissue. Okay, so here's one neuron, and you can see that it's long axon. It goes over here to the right. Okay, there's others here, but you can also see these smaller little kind of branching feeler things. If it's around this top cell body, those are called dendrites. I always find this stuff kind of interesting, where these particular cells have a lot of information that they have to process. The little dendrites that come around the cell body, that's all incoming information. So it hooks up, those hook up to all of these other neural cells around them. And those things are sending information out. So this one cell has all of these dendrites with incoming information, and it has to process that information and then possibly send out that same information. So if, and we're not going to get into that in specifics, but it has to meet some electrochemical threshold. So the whole process seems like it would have a mind of its own and thinking. It doesn't. It all has to do with chemistry and electrical charge. And if things change enough to produce some big enough charge, then it will send that same chemical message down its axon. So kind of interesting. There's also other, in one tissue, you don't have just one type of cell. Now up here, they're not showing you anything but neurons. There are other types of cells in our brain called neuroglia. They're usually very, very small, and they will sit in and around these neurons. They have, for a long time, they thought they didn't really do much. They were supportive. So they just help to nourish the neurons. But now they know that neuroglia actually also have some conducting mechanism. Get all of those together, we have an organ. So here they're showing you a brain of a goose. An organ system is all the organs that have a similar function. The nervous system, while it is very difficult to understand exactly how it's functioning, the brain, of course, we still are way from understanding every part and how things work. But the system itself doesn't have a lot of parts. You have the brain, the spinal cord, and the nerves. Above that, if we look at all the systems together, we have the organism, so we're seeing the full Canada goose there. Then we have a population level. A population can be defined like this: same species, same place, same time. Population. Same species, same place, same time. So it's exactly what that says. It's all of one species that are living together in the same place at the same time. Obviously, we can't include some goose into that population that died 20 years ago. Okay, so it's currently what we're seeing. Populations can also be difficult to try and tell one from another, because you've got a whole bunch of Canada geese. Are you talking about all of them that just kind of hang out in one pond? Or are we talking about all of them in the county? Are you talking about, what are we talking about? For sexually reproducing organisms, it's usually those that tend to stay together and reproduce with one another. But there's always some overlap, where if you have two populations that are existing kind of near one another, you may have members of each going back and forth. White-tailed deer is a good example of that, where they roam pretty far, so where do you have the end of one population and the beginning of another? It's not that clear-cut usually. Above population, we have species. That would be all of the Canada geese in the whole world, or all of the white-tailed deer, all of, you know, a certain type of bacteria, whatever. They go back then to community. This is back to the same place. A community is all living things living in the same place at the same time. So in this picture, they're showing the Canada geese here on a lake or a pond, but it's everything else that would be living here too. So all of the plants, both in the water and out, all of the other animals, all of the bacteria, all of the fish, all of the archaea, everything that's alive is included in the community. And every organism in a community typically has what we call a niche. They have some role that they fill. For example, we have certain types of creatures that decompose dead stuff. If we didn't, imagine how much trash we'd end up having. You know, what happens to all the leaves every fall? Even those that are not raked, say in a forest. Well, they decompose, right? That produces nutrients to go back into the soil, and then that gets reused in the plants. So that every year it's kind of a cycle where the leaves are shed, they fall down, decompose, the nutrients are there to be used by the trees, the plants, the fungi. So the decomposers are something that a lot of times we don't see. It includes fungi, but it also includes a lot of single-celled things. So everything has some role to play. It's not always pleasant, like if you watch any nature type shows, they might show you, lions when they hunt. So it's not always pleasant to watch some of those things, but that's nature, and it has to happen that way. The lions, they have to eat too. All right. And then above community is ecosystem. This is where we get to the living and the non-living. One ecosystem includes everything living plus all of the non-living components, like temperature, water availability, humidity in the air, amount of sunlight. So all of those things go into one ecosystem. That's usually what dictates how much energy is available to anything living, and thus what types of living creatures are there and how many. So for example, this ecosystem that they're showing looks like, I can't tell if this is part of the mountain system or if it's in the distance, but it's some aquatic environment with, looks like lots of plant materials, et cetera, that's pretty rich ecosystem versus, say, a tundra or other dry desert type ecosystem. If there's not a lot of water, then usually that'll cut way back on how many species are there and their numbers, because water is usually necessary for anything to be alive. Usually, not always. All right. Emergent properties. It's exactly what it sounds like. These are properties that you don't see at lower levels, but as you go higher in the hierarchy, emerge as something new. It depends on the lower levels, though. Two examples here that they have are metabolism and consciousness. What's metabolism? Yeah, it's how you process energy. In a cell, we can talk about cellular metabolism. It just means all of the chemical reactions that happen within a given length of time. That's the metabolism of the cell. So here what they're saying is, well, at what point then could we call that a property? In a cell we can, but if we start dissecting the cell, going backwards, we would have macromolecules. Would that be metabolism? Not if you're looking at just one. You have to have multiple so that they're interacting with one another. So if I were classifying, I'd say that that's what emerges when you go from the macromolecules, the organelles, up to the cell. Now, certain organelles, you could argue that they have their own metabolism. So then we'd have to step back one further. Consciousness is another one. If you were making a human or something living from scratch that has can be conscious of themselves, at what point would they be conscious? Would it be when you put the brain together? I don't know. That's hard to say. Is it at organ system or is it at organism? But that's kind of the point, is that if you look at the individual pieces, molecules aren't conscious. So it has to be above that. All right, biological themes. You don't have to write a lot of these down. This is just going to give you an idea of what you'll encounter this semester and how these things tend to be linked together. So they narrow it down to 5. There are lots of different themes, but they're just going to give us 5. And you'll hear about these things repeatedly, even if we're not studying in that exact chapter. Like evolution, we're talking about it a little bit now, but we don't actually get to the chapter on evolution until the very end. But since all living things evolve, then it's going to run through all of the chapters. Evolution is just considered change over time. Change over time. The guy, when we get there, the guy that we'll study as the father of evolution is Charles Darwin. He is not the first one that thought of the process of evolution. He thought of how does it work. The thought that evolution occurs came before Darwin, because long before him, people were noticing fossils in rocks and how there isn't anything like that alive that they can find. but maybe there are similar species, fossilized bones, that type of thing. So they had found those already. So the thought that things evolve was already kind of current, but no one had given an explanation for how that happens. And that's what Darwin did. His response, his answer, is what he called natural selection. Natural selection is survival of the fittest, not necessarily meaning the fastest or the smartest, but any organism is considered to have high fitness if it can survive and reproduce well. Name something.
Cow.
A cow, a rabbit, mice, bacteria, especially things that can reproduce into big numbers quickly, their fitness is really high. Okay? But cows and other mammals also tend to be high. We won't get into it in here, but if you guys go on through science and you take an ecology class, you'll talk about different life strategies, meaning different strategies to ensure that their young survive. So on one hand, mice, rabbits, a lot of trees and plants, they have a strategy where they produce enormous numbers of offspring. Why would they do that? Exactly, because a lot of them are going to get picked off and die somehow. But if you produce lots of offspring, the chances of one or more surviving is pretty good. Those types of organisms usually are not really in contact parents. Okay? Some of them might be parents for a short amount of time, like rabbits. Others, not at all. Turtles, for example. They lay their eggs and then they leave. Okay? So they're not in contact with them at all. But they usually lay big numbers of eggs. So if you've ever seen sea turtles hatching, they'll show that there's huge numbers that hatch usually all around the same day. And there's big numbers. It's like a feast for any of the nearby animals, birds, that type of thing. So a lot of them will get eaten. And if they don't get eaten before they make it to the water, then they're eaten when they get there, because you'll also have a bunch of carnivorous fish that will feed on them. But there's so many, there are some that will survive. So that's one type of life strategy. Another type is the opposite. Like humans, like cows, like elephants, anything that you can think of that only has a few. And we typically have one, maybe two, occasionally three naturally, right? Or I don't know if I've heard of anybody naturally conceiving more than that, but elephants are the same way, usually one. But when we compare, in terms of time spent parenting, we're talking about years. And so the idea is there's a lot of energy put into, one or two offspring and raising them, where for years they stay with you. And as a parent, then you're ensuring their fate, more or less. Does it work better than the other? I don't know. I mean, for us, obviously it does. But when you compare all species, if you take your own personal thoughts out of it in being a human and you just look at it from an objective point of view, you could make the case for either. One other thing about evolution before I go on. Evolution is a scientific process. It doesn't mean that you personally have to go with it in terms of your religious beliefs. It's scientific. Religion is not scientific. What's the difference? Anything that's scientific can be tested. We can't test for God. We can't test for ghosts. So those things aren't considered science. What you'll see on TV or read is usually not the general agreement of all the scientists. They will find usually the most extreme, and that's who they're going to interview, because it makes for better press coverage, makes it more exciting, and the ratings go up, which is unfortunate because they're supposed to be unbiased. And it's not that they're being biased necessarily by the interview, but they're being biased in whom they choose if they're doing it on purpose. All right, the flow of energy. Well, we know that all living things require energy. Ultimately on Earth, our energy comes from the sun. How do we capture it? What types of organisms? Plants. Usually that's where it starts. There's a few photosynthetic, say bacteria, archaea type organisms, but usually we're talking about plants. And so they're able to capture that energy, and then thus starts the food web. So then other things eat them, other things eat the things that eat the plants, so on and so forth. And so that's the flow of energy. But when energy is transferred, so when the plants grab energy from the sun, they don't grab 100% of it. And when the cow eats the grass or the hay, the cow can't capture 100% even in what it eats. There's always some that's going to be lost two ways. One is that the bonds can't be broken because there aren't the correct enzymes or something. And two is that means some energy is still left in poop. Well, that's why we have things like dung beetles. There's still energy in the poop, so that's what they feed on. The other reason is that especially for warm-blooded organisms, and the cold-blooded ones too, but they get heat from the breakdown of the food. Well, that heat is actually coming as a loss of energy. Now, for those of us that are warm-blooded, it's a good thing because that helps us to keep our body temperature where it is. But that's where it's coming from. Your car, same thing. You pay for gas, you put it in the tank of the car because your car requires energy, but it doesn't get to use all of the energy in the gas. It uses some to move the parts. Some of it goes out the tailpipe because it doesn't break every bond. And some of it is lost as heat. Just the way it is. And as I said before, if you look at various ecosystems, how much energy is available, that's what's going to determine how diverse the ecosystem is. Cooperation, this is also something that's interesting to study, is cooperation between different organisms, different species. It can be something that's kind of warm and fuzzy where they both benefit from it. Okay? Like, for example, have you ever seen the little birds that ride on the top and backs of hippos or rhinoceros? Okay, so that's called, I think it's called mutualism. Anyways, it's mutualistic because they're both getting something. So what are those birds eating? Why are they on there? Yep, flies, ticks, that type of thing that bother the hippos, the rhinos, that type of thing. So the birds are getting a food source. The hippos and rhinos are getting their skin cleaned. So that's mutual. That's a good thing. There's another one that I think is called commensalism. Might have that backwards, but I think that's right. One benefits, the other one's not harmed. So it doesn't really matter to the one that doesn't benefit. It's not being harmed either, but one will benefit. And then there's a third type called parasitism. You guys probably are aware of that. What's a parasite? Yeah. One benefits the worm, the other one is harmed. Okay, so like a tapeworm, like ticks, like certain bacteria, viruses, although we don't consider them truly living. Symbiosis, that's when two species live in direct contact. For us, we have all kinds of organisms that live in and on us. Okay, so it could be bacteria. In some cases, parasites, bed bugs, scabies, certain mites, lice, that type of thing, which I know is kind of gross, but that is, even though it's not a nice, warm and fuzzy relationship, you still consider that symbiosis. Number 4, structure determines function. You'll hear that time and time again, and especially if you go into A&P and you take that class, you'll hear that there too. But when we look at, say, cells, how they're different in shape, that neuron has that cell body with all of the extensions and then the very long axon tail. Its structure is linked very much to its function, what it does. It has to have all the extensions so that it can gather incoming information. And then it has to have the long tail so that it can transport a message. sometimes far away to, say, your leg muscle. Some of those axons are really, really long. You know, the ones that innervate your leg muscles, they typically come directly from your spinal cord. So they're long enough that you could dissect and hold one up, you'd be able to see it. Very, very long. Other structure and function. Your muscle cells have long fibers, and then they have these little things that stick out that look like golf club heads. And what that's for is to shorten the muscles. So they kind of hook together like this. When your muscle is stretched out, everything's kind of laying flat. But when you contract a muscle, you're trying to move two bone parts, and you need to make that area shorter. And so then they move like this. And so again, we can see that the structure is very linked to what it does. So that when scientists are viewing something new creature, new type of cell, whatever, that's one of the things they look at. Because one of the questions is going to be, what does it do? What's this part for? Maybe an old fossil where we've never seen that type of bone or that type of structure. What was it for? And so that's what they use. They try and figure out what the function might be based on what its structure is, and also research maybe other creatures that are similar. Adaptation, this has to do with evolution. But basically, the thought for adaptations is that when new structures evolve, they evolve in an adaptive manner, meaning to help that organism live in that environment. So it explains why things that live in the water, they have flippers or webbing. Why? Yeah, to get around through the water, right? So that they can get away from predators, or they can be better at getting down to the bottom to get their food, or whatever it might be. And so evolution tends to work that way. It is not biased. Okay? So we can't think that maybe Mother Nature chooses one structure over another. It's not so much a choice as it is just that whichever organisms are best suited are the ones that reproduce more and survive better. Number 5, homeostasis. We already talked about that. So that's steady state, remember. And a lot of the energy required for a living organism goes into that. This is constantly being maintained. Even in single-celled organisms, they're constantly maintaining what's inside the cell. So there are, for example, little protists, single-celled eukaryotes, that have this vacuole that's called a contractile vacuole. And it's for contracting the whole cell to wring it out. If you ever wring out a washcloth, you're trying to get rid of the excess water, that's what the contractile vacuole does. Because the cell on the inside is saltier than the water on the outside in the lake or the pond or wherever. So water tends to leak in through the cell membrane. If that organism doesn't do something about it, it's going to die, because the whole cell is going to pop. All right, stages of a scientific investigation. They're going right into the scientific method. Okay. Scientists, no matter what field they're studying, always use the same set of steps for some sort of investigation. It could be biology, it could be chemistry, it could be physics, it doesn't matter. They always use the same set of steps. Now, I'm going to give you 5, but it actually varies from one book to another. I've seen as few as 5, which is why I stick to 5, because why memorize more if you don't have to, up to like 10 different steps. The more steps there are, it just means that they're subdividing the 5 main steps more. So we're just going to go through 5. The scientific method, just by definition, this is a systematic approach to discovering new information. What does systematic mean? When somebody does something systematically, what are they doing? Doing it the same every time. This decreases mistakes. That's why they do this. It also decreases mistakes in translation so that we have one group of scientists studying something on one side of the world, and you can have another group studying the same thing, and they can share materials and information. Especially if it's done systematically, then it's much easier to understand and with fewer mistakes. So number one is observation. Make sure for the test you can list these and define each step. Number one is observation. Well, of course it's observing. You know, if you find something brand new, one of the first things you're going to do, of course, is observe. Where did I find it? What color is it? What does it seem to be feeding on? At what temperature? How much sunlight? All of that. But also research. Research for any extra information you can find. If it truly is like a brand new species, you can't research that species because nobody knew about it, but you can research similar species or similar environments. Okay, so, I don't know, it's been like 10 years ago. It hasn't been too long. There was a group of scientists that were exploring a cave in California that had never been explored, which is kind of a shock in today's world, but they found a whole group of brand new species of cave organisms. And that was big news at that time because it hasn't happened in this country for a long time. So they would go through the scientific method, and they're going to observe first, and then they're also going to do background research. What other types of cave organisms are like these? And that might help them to answer the questions of what are they feeding on, how do they reproduce, etc. Number two, that's the asking questions part that I just went through. So they're going to list all of the questions they have that can't be answered by what they already know. Number three, form a hypothesis. What's a hypothesis? Educated guess. They try to answer their own questions. So with observe and research, you already know quite a bit. You try to answer your questions based on what you know, what's logical. This part, the hypothesis, can be kind of tricky because you want to make sure that a hypothesis is testable. It's not good if it's not testable. Why is it not testable? Many times it could be from lack of technology. For example, going down into the very deepest parts of the ocean. Have we been there? Nope, because we don't have the technology to go down there yet. The pressures are way too great. And so we've been sending probes and other things down, but it sure would be nice if we can get a group of scientists that can go down there and really take some samples and see what it's like down there. So until then, until we get that technology, we can't go past a hypothesis. Other times, we might have the technology, but if it's a poor hypothesis, then it's not testable. So this is also, for example, why religion and the supernatural aren't part of science, because we have no hypothesis that truly is testable. Okay, so we can make hypotheses on, ghosts. There's nothing I can test. So scientists shouldn't say whether they do or don't exist, because that's their own personal belief. It's not testable. Same thing with God. That's their own personal belief. There's nothing that we've devised thus far that can test for whether God exists. All right. Alternative hypotheses, there might not be just one. So they might have several possible explanations. Number 4, testing. Okay, well, this is where an experiment is actually done. This may well be the very longest part. And really, this is one of the longest parts, is not just during the experiment, but it's actually setting it up. Scientists have to be very, very, very picky and careful about this, because ultimately, the outcome is to publish something, share your knowledge with the rest of the world. But in order to get it published, scientists will submit to academic journals. And they have a committee of scientists that will nitpick and go through their paper. Not just looking for, say, spelling mistakes. They're looking for design flaws in the experiment. And as a scientist, that's the last thing you want, because it's humiliating, and you can also lose your job, because if you spend all of that time and money, It doesn't matter if you're talking about working at a university or working for a company. They're not going to be very happy with you if you went through all of that time and money and it had a big flaw, so it's not worth anything. It means all your data and results don't mean squat, and you would have to start over. So they're very careful about how they set these up to make sure that it is a good, objective experiment. To do that, a lot of times they will use a control and variables. It's exactly what it sounds like. A control is something that doesn't change, it's controlled. Ideally, with science, you can bring it into a lab and set it up, but that doesn't always happen. You know, if you're a meteorologist and you're studying hurricanes, you can't bring a hurricane inside. You can try to simulate, can't bring it inside. Same thing with volcanologists. They study volcanoes, can't bring one inside. So okay, variables are those things that vary or change. In an ideal experiment, you have one or more controls. Controls are always for comparison, so that you can tell if there's a change. And then you test only one variable at a time. A good example is if we were studying why mussels in the Missouri River are declining in number. So mussels like clams, they're not exactly the same, but okay. So they are in decline. If I ask you guys why, I bet you could come up with some reasons. Why would you think they're declining? Okay, well, first of all, where do they live? In the river, but where in the river? On the bottom. Okay, they can't move around very well. Okay, so they live on the bottom. What else do you find in the bottom of the river? Mud and sediment, that type of thing. Other carnivores maybe. What about any pollutants? If they're solids, yes, you know, and they sink. So now give me some reasons they might be in decline. Do you think that they need access to the water? Probably, right? So what if they get buried in sediment? It's probably not good. And so that might be one reason. Another reason might be pollution. Now, what pollution? That would be much more difficult to answer. Another reason we might come up with is, let's see, there's no light down there. Do they need light? I don't know. Maybe they do. But because the water is so murky and there's so much sediment in the water, there's no light getting all the way down there. What about zebra mussels? Have you heard of those? Zebra mussels are an Asian type mussel that was brought in by accident in hulls of huge cargo ships that ultimately by the St. Lawrence Seaway can actually make it all the way to Chicago, so through Lake Michigan. And they're small, so they hitched a ride and then they got into Lake Michigan. They reproduce in huge numbers and they stick to a lot of our native mussels. So some of our mussels in the Mississippi River can be pretty big as my hand or bigger, and it'll be covered with all these tiny little zebra mussels, which means that they're kind of suffocating it. They're getting all of the food, and all of that weight is on top of that shell. And so the mussel can't open as it needs to filter feed. Okay, so maybe that's why. There are laws in place now where if you were to grab one of our mussels and it's covered with zebra mussels, they actually want you to scrape them off and throw them up on the bank, kill them. Because we haven't found anything yet that can target just the zebra mussels. So there's still a big problem for us. At any rate, if we were going to test that type of thing, we could bring that inside into different fish tanks, aquariums. So the control is the environment of the aquarium. We can control the temperature, we can control the amount of light, we can control the amount of food, so we can keep the environment whatever we want in those aquariums. So we could have a positive control, and that's going to be like the perfect nirvana for the mussels. Everything's going to be great in there. We're going to see how well they reproduce. And we're probably only going to do one species at a time, unless we find that one or more species have to live in contact with one another. That's a possibility. But you don't want to add too much to muck up the results. Then in another tank, we're going to introduce one variable. So we found mercury in the water, let's say. So we're going to put mercury in that tank and see what happens. In another tank, we're going to put a bunch of silt in and bury those mussels and see what happens. In another tank, we're going to put a dark cover it or put it in the closet to make sure it doesn't get any light and see what happens. So you guys get the idea that we're only testing one thing at a time. That way, when we find that the one in the closet is doing just fine, we know it has nothing to do with the lack of sunlight, so we can rule that out. And so that way we start to narrow down what the possible hypotheses left, what's still possible. So that would be a good design. We can't do that with everything, and sometimes we don't want to. For example, if you're working for a company in development, when they go to sell things, the people they're selling to don't want to know what happened just in a controlled lab. Do you want to take pharmaceuticals that haven't been tested on people? I don't. And so that's something that we don't want just in a lab. It'll start in a lab with, say, rats or rabbits or monkeys or some sort of mammal, which I know some people don't like. I'm not a fan necessarily. I wouldn't want to be the one that's actually doing those experiments because it would make me sad. But how else are we going to test it without just trying to get some human people? And you can't do that. So it has to pass so many trials first. If you're a farmer and somebody wants to sell you some new herbicide, but it hasn't been tested on crops outside, do you want to buy it? No. So again, they develop it maybe in a lab, but when it's ready for trial phase, they take it out to a for real plot. And so initially in the greenhouse, maybe it works great, but then you take it outside to see what Mother Nature has to offer, and it doesn't work so well, then you have to figure out why. All right. Number 5 is conclusion. Okay. Notice that it says retest. A lot of times that happens automatically, and it doesn't matter if your hypothesis was supported or not. If your hypothesis is supported, if it's the first time it's ever been tested, then, I mean, that's great, but does that make you sure that that's what the real cause is? No. It's kind of like pharmaceuticals. We don't want it tested just once. We want it tested many, many, many times before we take it, before it's released to the public. If the hypothesis was wrong, well, then typically that scientist or group of scientists will want to go back and retest maybe a different hypothesis. The most important part of this is to publish. If scientists don't publish their work, then it's not shared with anyone. And so then if I were to assign research papers for you guys, you wouldn't have anything to research. Then maybe somebody's blog, Wikipedia, and those don't count. Okay, so when they publish, that's why a lot of times when you're assigned a research paper, it'll say so many sources from peer-reviewed sites or journals. Peer-reviewed is exactly what it sounds like. It means your peers in your same profession are going to review your paper. One of the things that they test for is, of course, experimental design, but then there's all kinds of other things that you'll have to go through if you're trying to submit for publication. Depends on what journal you're talking about. Some are much more prestigious than others. The ones that are much more prestigious, it's because the scientists on those panels are much pickier. There's always a length. You know, the papers that they publish, if you think about like doctoral dissertations and stuff, it's a book. You can't put a book in a journal, and so they're going to tell you, okay, you have to whittle it down to 5 pages. I mean, that's going to be really hard to do, and so they go back and forth, trying to get something that will please that peer set before it's actually approved for publishing. And sometimes it never is. And so then you just have to go find maybe some other journal And so you kind of go down in rank in terms of which journal will publish. But publishing is really important. This just shows a flow chart. Here they're showing you 5 hypotheses. That's fine. They test them separately. It shows that whatever 1 and 4 were, let's say, are tanks. So 2 of the tanks we can automatically see they're doing just fine. So we reject those. Then we test 2, 3, and 5 some more. We can reject 2 and 3. That leaves us with 5. Okay, so hypothesis 5 looks like it's going to be the one. It's not proven. It's supported. In science, you should never hear them in a paper say that it's proven. It's fact, because everything is always open to more testing. We don't know if something's going to change in the future. You know, what if gravity changes? Okay, so we'll talk about that. Theory, law, and certainty. So that's this next part. I don't consider theory and law to be part of the scientific method because it's rare. And you guys are familiar with some of these. If I say theory of, what do you say? Evolution, relativity. We'll talk about the cell theory on the chapter on cells. And there are genetics theories. So a theory in science is a very big deal. In everyday language, it's not. If I tell you that I have a theory about why they broke up, that means I don't really know. In science, if there's a theory, it means the scientific community, most of them, if not all of them, believe that that's the truth, that that's what's happening, whatever that is. These are usually really broad. You like theory of evolution. We're talking about all living things, not just the evolution of pigs. Okay, so theories are very big, very broad. They have been around for probably hundreds of years and have always been supported. So time after time after time, they've been tested and tested and tested and tested, always supported. So in terms of a definition for a theory, notice here it says, this is really big, long, a degree of certainty, unifying explanation, yada, yada. It's not one hypothesis. It's a set of related hypotheses that have been supported by testing for at least tens of years, if not hundreds of years. Okay, so they've been around for a long, long time. Usually if it gets to a theory, it means the scientific community mostly is in agreement. The general public, maybe not yet. It depends on what the topic is. So later we'll talk about how some of the responses from people is it's just a theory. In science, that's not a valid argument because a scientific theory means that there's an awful lot of data and evidence to back it up. Finally, a law, this is as high as you can go. A scientific law is a theory that's probably hundreds of years old. It's old. This stuff has been around for a long, long time. The evidence, there's so much that it's almost undeniable. This is when the scientific community, all the scientists pretty much believe it, and probably the general public does too. That's a scientific law. It says highest degree of certainty. It still doesn't mean it's proven, and it's not a fact. But it's as sure as we can get. Like the law of conservation, the law of conservation of energy, gravity, or conservation of matter, whichever. The law of gravity is a good one, where we're sure that if we drop a rock, it's always going to fall to the surface, as long as there isn't something else that could explain it away, like massive wind or something. But I can't tell you that it's going to be that way forever, because what if gravity changes? That's why we don't say it's proven or it's a fact. They are always open to being tested further. So if somebody thinks that the law of gravity is not correct, they could test it again. In order to convince everybody that's not correct, they're going to have to test and retest and retest to show with evidence why it's not correct. theory law says goes through just what I said. It's provisional. We think it's correct until it's proven otherwise. You can disprove something. There's lots of evidence that disproves a hypothesis or a theory, even a law. That doesn't happen very often. Judgment and intuition. Here they're talking about, well, how do we know when something becomes a theory or a law? It's not black and white that way. So there's not necessarily a panel of scientists that sits around and try and decide, are we going to make this a theory or not? It's not, kind of not like that. Same thing with theory becoming a law. It's something that kind of gradually either is accepted or not. I mean, for something to become a theory, the scope has to be really, really broad. And when I did my master's thesis, it was very narrow. When you do thesis type work, either for a master's or for a doctorate degree, it's kind of intimidating at first because they tell you that whatever you're doing for research has to be absolutely authentic and no one has ever done it before. You know, at first that's kind of like, oh my gosh, well, how am I going to come up with something that original? But it's something like this. I studied the growth rate of red-eared sliders, that's a type of turtle, in the Mississippi River pools 19, 20, and 21. That's how I got it to be original, is because nobody studied that in the Mississippi River pools 19, 20, and 21. So that's a lot of times what happens. But that adds to the growing group of studies that have been done on not just red-eared sliders, but other freshwater turtles. And then once we're talking about all freshwater turtles, we might be able to include all seawater type turtles, saltwater. You get the idea. But to become a theory, it's going to have to get bigger and bigger than that. Like the theory of evolution. That applies to all living organisms. And then for it to become a law, that's going to happen when it comes right down to it, when somebody is brave enough in writing to call it the law of evolution. If the scientific community is ready for that, then nobody will really say much and it'll pass by all right, and then other people will start calling it the law of evolution. If they're not ready for it, then whoever wrote it is going to be absolutely raked over the coals and scathed for, you know, jumping ahead and calling it the law. So that's kind of where we're at with that. And then finally here they just talk about how we can't test everything, and I already talked to you guys about that. Okay, so it has limitations because some things can't be tested. Practical limits, like you can't take a volcano into a lab. All right. We'll cover the rest of it next week. I'm sorry, on Wednesday, when I see you guys. Make sure that you have read chapter 1 and chapter 2. If you don't get all the way through chapter 2, that's all right. I won't cover it all on Wednesday, but I'm going to start it. I think that's it. See you guys Wednesday.
Mr.